Hello everyone, you are welcome to Chemistry 318. We are going to start from where we stopped. We stopped in the last class when we considered the mechanisms of interaction of electromagnetic radiation with matter. In the last class, we looked at what electromagnetic radiations are. We look at the different regions that makes up the electromagnetic radiation. We talk about the interaction between matter and electromagnetic radiation. And we said that the study of the interaction is what we call the spectroscopy, which is the basis of my own aspect of 318. Then we looked at the different characteristics of electromagnetic radiations. We said they are waves, they behave like wave, they have they have the electric and the magnetic field components, and we say they travel perpendicular to one another. Then we talked about the energy of an electromagnetic radiation, which is given by E equals to hv, which is equals to hc over lambda, where h is the Planck's constant, E is the energy, c is the speed of light, while lambda is the wavelength. And that the electromagnetic radiation is divided into different regions. We have the gamma, the IR, the UV, the visible in between the IR and the UV. We have the gamma, the X-ray, the UV, that is the ultraviolet. We have the visible in between the ultraviolet and the IR. We have the microwave as well as the radio waves. And we said that the region of the electromagnetic radiation where the eyes can see what makes up that region is the visible spectrum. The visible spectrum is that region in the electromagnetic radiation where human eyes can easily see, and we can differentiate between violet and red, and that the wavelength of that region is between 400 and 780 nanometer. So later we moved on to chromophores, and we said chromophores are what the molecules contains, which makes transition between energy states possible. If there are no chromophores, we are not likely to identify or to see any intensity of absorption. And generally speaking, transitions that are favored in quantum mechanical terms are the ones that exhibit absorption bands, as we know that there is possibility of transition from lower energy level or ground state to the higher energy level or excited state. And transition can be from ground state molecular orbitals, that is the sigma bonding, the pi bonding, and the non-bonding, and to excited states, which are sigma star and antibonding, sigma star and pi star antibonding. All these transitions are observed in the UV region. When you talk about molecules can possess energies in different forms from what we know. Energy can be vibrational, rotational, or electronic. So transition can be from one energy level to another, either after absorbing or emitting photons of energy. So if there is transition from lower energy level to higher energy level, that means there is absorption of photons. And at the higher energy level, if the molecule is not stable, it wants to attain a state of stability, and in such doing, it can move back to the lower energy level or even to ground state by emitting the photon that was initially absorbed. And we later talked about electromagnetic spectrum and the processes that occur between the electromagnetic spectrum and the molecules. We say that different processes occur, that molecules have the tendency to undergo different processes like rotation, vibration, electronic transition, and nuclear transitions after absorbing electromagnetic after absorbing photons. So the energies underlying these processes are found at different regions in the electromagnetic spectrum. The energy that is involved in the processes which molecules undergo are found in different regions of the electromagnetic spectrum. When it is radio waves, that means it is the radio frequency, it is in the radio frequency region. And radio frequency region has very low energy that correspond to the energy differences in nuclear and electron spin states. This particular energy in question, or the radio frequency waves we are talking about, which occurred in the radio frequency region, find applications in nuclear magnetic resonance (NMR) and electroparamagnetic resonance spectroscopy. Then after radio frequency, we have the microwaves. The microwaves have energies between those of radio frequency and the IR, that is infrared waves, and it finds application in rotational spectroscopy and electron paramagnetic resonance spectroscopy. Then we have the infrared radiation. The energies are associated with molecular vibrations in the infrared region of electromagnetic spectrum. So when you talk about infrared, it is associated with molecular vibrations. So infrared spectroscopy is also known as vibrational spectroscopy, and it's very useful in technique for functional group identification, mostly in organic compounds. You want to see the kind of functional groups that your known compounds contains. Maybe it has a carboxylic group or a keto group or what have you. So those functional groups, a double bond. So those functional groups, we all know what functional groups we are talking about. Functional groups are the substituents on the backbone of your molecule. So it can be a carboxylic group, it can be an amine group, it can be amide, it can be keto, it can be aldehyde, and so on and so forth. Then from the infrared region, we have the UV visible. In between the infrared and the ultraviolet region, we have the visible region. And the visible region, as I've said earlier, is the one that human eyes can see and differentiate different colors. So the UV visible regions are involved in electronic transitions in the molecules, and it found applications in methods such as UV or visible light under electronic spectroscopy. Then we have the X-ray radiation, and the X-rays are high energy electromagnetic radiation that causes transitions in the internal electrons of the molecule. So when you subject matter or substance to X-ray, you are bombarding it with high energy radiation, and it can now causes transitions in the internal electron within the molecules. Then we move on to mechanisms of interaction. How does the interaction occurs between matter and electromagnetic radiation? For any molecule to interact with electromagnetic radiation, that means the molecule must have some electric or magnetic effect that could influence that could influence the components in the radiation. What I'm saying is, before an interaction can occur, from what we know, it means from the electromagnetic radiation point of view, your electromagnetic radiation contains both electrical and magnetic fields. Likewise, for that matter to be able to interact with the electromagnetic radiation, that means it must also contain some electric or magnetic field. Let's take, for example, in NMR, nuclear magnetic resonance spectroscopy, the nuclear spins in a molecule have magnetic dipoles which are aligned with that of the radiation. The NMR, in the NMR, the molecule too must have nuclear spins so that there can be interaction between your NMR and the molecule. Then, interaction with radio frequency of appropriate energy also results in change in dipole, because you know that once you subject your matter or substance to electromagnetic radiation, there's going to be an interaction at a particular region or in a particular region of the electromagnetic spectrum. Then we talk about rotations of a molecule having a net electric dipole moment. So your molecule must have a net electric dipole moment before you can talk of interacting or interaction between the molecule and electromagnetic radiation. The vibrations of molecule also can result in changes in electric dipoles that will interact with the components in the electromagnetic radiation. Electronic transitions can also take place from one orbital to another. So electric or magnetic dipole moment in a molecule is required for absorption or emission of electromagnetic radiation. What am I saying there? Before you can have interaction between matter and your electromagnetic radiation, that means the molecule must either have electric or magnetic dipole moments. This is very key. It is required in a molecule before you can talk of absorption or emission of electromagnetic radiation. From what we know, like dissolves likes. So for electromagnetic radiation to interact with your molecule, remember that the electromagnetic radiation has magnetic and electric components. Likewise, your molecule must have a magnetic or electric dipole moment before an interaction can occur. The interaction can now be either absorption or emission, depending on the molecule in question. Then I move on to laws. There are laws that guide the absorption of electromagnetic radiation. So we talk about laws of photometry. Laws of photometry, we've all heard about Beer-Lambert's law. Lambert's law and Beer's law. So combining the two together gave what we now called Beer-Lambert's law. And what does Lambert's law says? Lambert's law says that equal parts, that absorption states that equal parts in the same absorbing medium absorb equal fractions of the light that enters them. What we are saying there is that when you have a molecule, equal parts of the molecule in a medium will absorb equal amount of light that goes through them. Whereas Beer's law states that the absorptive capacity of a dissolved substance is directly proportional to its concentration in a solution. So Beer's law talks about the absorptive capacity of a substance that is directly proportional to its concentration. The capacity of the substance to be able to absorb light depends on the concentration in solution. So let's say, for example, you have a known amount of substance and you allow light to pass through the substance, the molecules of the substance will absorb part of the light that passes through them and allow the rest to just pass by. Remember that there are molecules in your solution, so depending on the amount of the solute or the dissolved substances or the dissolved substance in the solution, it will determine the amount of light to be absorbed while the rest is being allowed to pass. So invariably, there is a mathematical expression to to express what we are talking, what Beer and Lambert's law is talking about. And it says that if an incident radiation of the resonance frequency having intensity of I naught enters a sample cell, its intensity decreases as it penetrates the sample. What we are saying is you have a beaker that contains solution of a particular substance. As light passes through the beaker, some of the light will be absorbed by the solute in solution in such a way that as the light penetrates through the sample, some of the lights are being absorbed, which now results in decrease of intensity. The intensity of the light that enter the samples and passes through the samples will be decreased from what goes in at the point of entry to what is being released after some of the lights are being absorbed. So we say mathematically, the Lambert law says that absorbance equals to epsilon C L. That is epsilon multiplied by concentration multiplied by the path length of the sample cell. A is the absorbance, which equals to the molar concentration C. The path length is L, and the epsilon is the molar extinction coefficient. And molar extinction coefficient depends on the nature of the chemical and the wavelength of the light used. So A absorbance equals to epsilon CL. So that expression is known as Beer-Lambert's law, or let me say the relationship between absorbance and the concentration of the absorbing species is known as the Beer-Lambert's law. Remember that before you can talk of absorption, because before some of the light can be absorbed, that means the molecule contains what we call chromophore, because that chromophore have the tendency, and it is that part of the molecule that actually allow absorption to be able to be possible. Without chromophore, that means the molecule or substance will not be able to absorb any light. I want you to, at this point, read more on chromophores. Try to understand what they are, understand their characteristics, so that you will have better knowledge and better understanding of what Beer-Lambert's law is saying. So Beer-Lambert's law, the expression for Beer-Lambert's law is A equals to epsilon multiplied by concentration multiplied by the path length of the sample cell. Then we talk about transmittance. Transmittance is another way of describing the absorption of light. Initially, we talked about absorbance. Now we are talking about transmittance. And transmittance is simply the ratio of the intensity of the radiation transmitted through the sample to that of the incident radiation. So invariably, transmittance is expressed as a percentage, and it is I, that is the intensity that is transmitted over the incident radiation, the intensity of the incident radiation. The intensity of the incident radiation is represented by I naught, while the intensity of the transmitted radiation is represented by I or IT. So I subscript T over I subscript 0 multiplied by 100 gives transmittance. So absorbance and transmittance are both dimensionless. That is, they don't have any unit. They don't carry any unit. You know the concentration unit, you know the path length unit. You can easily determine the concentration, or you can easily determine the unit of your epsilon. But absorbance and transmittance are unitless. Thank you.